Hello, a very warm welcome to everyone joining us live from the seabed. We are currently on board the research vessel G.O. Sars for our expedition benchmark, benthic habitats of the Denmark Straits. We are currently in Icelandic waters, approximately 130 kilometers west from the northwest tip of Iceland, and we are directly on the Arctic Circle, about 66.3 degrees north. My name is Emmeline Broad. I'm a researcher based at the Zoological Society of London, and I'm joined by the leaders of this expedition, Dr. Chris Jessen, a research fellow at the Zoological Society of London, and Dr. Julian Burgos. We also have Steinum Olafsdottir. Both Julian and Steinum are researchers from the Marine and Freshwater Institute of Iceland. And additionally, we have Nanette Hammerkin Arbo joining us from the Greenland Institute of Natural Resources. Firstly, we would like to acknowledge our funders of this expedition, Eurofleets, for the use of the RV G.O. Sars and the remotely operated vehicle ÆGIR 6000. They are provided free of charge for this expedition as part of the Eurofleets Plus project funded by the European Union's H2020 Research and Innovation Programme under Grant Agreement 824077. A big thank you as well to the Marine Stewardship Council. This expedition will contribute towards research conducted by Chris and I, funded by the Ocean Stewardship Fund, documenting vulnerable marine ecosystems in Greenland. If you have any questions, please use the comments section on Facebook. We will do our best to answer as much as possible. And one person or object I need to introduce is ÆGIR 6000, our remotely operated vehicle, or ROV. Give us a wave. Chris, maybe you can tell us a little bit about the ROV and what we've been using it for.
Yeah, so the ROV is a remote camera system. So it has a total of 9 cameras, I think that was right, you guys told me. And we have propellers and robot arm and a little vacuum cleaner that you might even see later that allows us to take samples, and there's a storage area at the bottom. We're just panning around to see it. We can see a second arm and a little tray at the base, and a little bit later on we'll be collecting some samples and placing them in the drawer, and this will allow us to bring these samples up from 600 meters depth back up to the surface. You can see the little information bar at the top of the screen, and that gives us details about the position and the depth of the vehicle, so that we can exactly pinpoint where we are and where we're taking our samples from. We're in very deep and very cold waters. We just conducted a temperature profile here, and although the surface is just under 6 degrees, down at the seabed we're just below freezing, but the pressure means that this water isn't going to freeze. This water is kind of a current coming from the north, so this is cold Arctic water. It created a bit of a problem for us a couple of days ago. The currents at the seabed were so strong that we couldn't operate the ROV, but thankfully today we're all good, and we can see this beautiful seabed.
Cool, thank you. So Julian, perhaps you can explain a little bit what brings us to this beautiful region of the deep seabed.
Hi, yes. Well, thank you for organizing this live event. We are super excited to be able to share our observations from the seabed with everyone. So, as you said, now we are in Denmark Strait, what in Iceland we know as Greenland. That's the area between Iceland and Greenland. And it's an area that is very interesting for us. It has very dynamic water masses. As Chris says, here the cold water from the Arctic meets the less cold or warmer water from the North Atlantic. So there's a very big differences in water temperature near the bottom, very differences in the type of seabed that you can find, and the current speed. So then this means that the habitats that we found on the seabed are very different if we move in different places. So, but this area has not been studied very much. We have several sources of information telling us that the benthic communities, the communities on the seabed, are very diverse. But except for some video dives on the coast of Iceland and a few on the close to Greenland, most of the areas in Denmark Strait has never been filmed or photographed before. So basically now we are doing exploration in the most basic sense. We are actually seeing and describing these communities for the first time.
That's great, thank you very much. So generally, what are we expecting to find down here? I know that you have done some trawls before and some video work in areas similar to this. Do you feel that we're going to find habitats that are dominated by sponges and deep cold water corals as well?
Yes, that's one of our main points of interest, is habitats that are complex and they're dominated by sponges and corals. We have several sources of information showing us that in some places we could find some of these habitats, the kind of habitats that people refer to as vulnerable marine ecosystems. These are places where there's a lot of animals that could be damaged if we do, for example, bottom trawling. So it's very important for us and for the management, both in Iceland and Greenland, to be able to find where these very rich seabed areas are, so we can work to protect them. So, and we're lucky because in this place we are right now, we have found a very diverse community of sponges and corals. The kind of thing that we selected the name Svamborg, which is sponge city, because of the number of species and the density that we see. And so we are very, very happy about finding this type of habitats.
So we're probably going to lift off in a second and have a bit more of a look around. As you can see, there's quite a dense community of dermosponge and soft cold water coral here. A number of dermosponge species and soft cold water coral species are protected under international law, and this is one of the things that can come out of work like this. So I think we're good to go.
Stop.
So we're just powering the ROV off the seabed now, and we're actually going to start to have a little look around this area. Chris, currently on the rock we've got a Gorgonocephalus. You know a little bit about this.
Yeah. So just to the right of the screen. The right of the screen there. We've got a basket star, and they're named after Greek mythology, the Gorgons. You might remember Clash of the Titans and the Medusa. So the name literally means dreaded head, and it's like snakes for hair, and it's a pretty cool animal. But how big? Anyone know how big they can get to? Yeah, maybe.
The disc can be over 10 centimeters.
Yeah.
10 centimeters.
The disc, yeah, and the arms, of course, will be
Yeah.
extended out on the disc.
Yeah. So. Yeah, so lots of people putting their arms out to me. So 50 centimeters?
Yeah.
Yeah.
50. Yeah.
Yeah.
It only has 5 arms, but each arms is like
Branches.
branching and branching again and branching again. That makes it like this basket form.
Yeah. So it's a relative of the starfish, hence the name basket star.
Yeah. We've got some really nice sponges underneath that. We actually managed to get a sample of those yesterday, didn't we, Skynum?
The white ones?
The white one underneath the
Yeah,
rock.
that's glass sponges. That's like vase form glass sponges. Quite. We haven't identified directly to species now.
Yeah, it's quite difficult to identify directly to species because a lot of the things that we're finding here are still relatively undescribed, or if they've been brought up using different methods, they don't tend to look like what they look like now.
Yeah.
Which is one of the benefits of using the ROV, or the remotely operated vehicle.
Yeah, most of the work that's been done on samples here are based on trawl surveys, and if you can imagine taking a large net and dragging it across the seabed here, everything's going to get totally bundled and squashed, and then brought up from 600 meters and the strong pressure there up to the surface. And so what we normally get to see when we're on deck and are pretty mangled and squashed specimens, and they don't retain the beauty, and often they don't retain the colours that you can see here. I mean, the really remarkable thing that kind of strikes me is when I'm looking at this incredibly dark environment is all the bright colours and the wide variety of colours and shapes that you can see on the seabed. I personally find it really amazing.
You can also, the advantage of having the video on the seafloor is that you can see the communities and how the different species are interacting with each other.
Well, we actually have a question about the community. We have a question that says, at what point would you call a community diverse, a certain number of species?
Yeah, that's a very good question. So, yeah, it's always a relative term to me. So it's not diverse versus non-diverse. It's a gradient. But this location has a very high number of species compared to other places that we've known. So.
And then I also think we should explain the two red dots on the screen, which is laser lights, and the distance between them are 10 centimeters.
And that enables us to measure the things that we can see in the screen. Thank you, Nanette.
Yeah.
Nanette, how does this look different to some of the imagery you've seen around the western coast of Greenland, or is it pretty similar?
Well, I think there's much more trawl fishery going on in western Greenland, but we have found habitats like this also in western Greenland, so it's not that different. So, but I think it often depends on what kind of sediment is there. So if you're having sessile organisms which has to be fastened to something like rocks, then you need rocks for them to be there. If you have mud ground, then you see another completely different habitat and with different organisms connected to it.
Yeah, a few days ago we did some surveys in Greenlandic waters on muddy seabed, and we saw some really nice sea pens and got some really amazing photographs of this very large sea pen called an umbellula, and the Greenlandic fishermen call them sea flowers. Is that right?
Yeah. Yeah, sea flowers.
Or in
Søblomster? No, I don't think that's what they call it.
No?
We've got a couple more questions coming through. Is there documented fishing / damage around this area that may endanger this section of the seabed?
No, there is not any fisheries going on in this particular place.
Yeah.
We have fisheries close to this, but it's a bit in a shallower ground than this one.
Is that because of legislation, or is that because of the rough terrain?
It's neither, actually. I think it's because the bottom was here is really cold, much colder than just if you're in like 200 meters depth just close by here.
Yeah.
this is the Arctic water mass dominating here, and that means that the fish that they are targeting,
Yeah.
it doesn't thrive, it doesn't want to be in this cold water. That's why they don't go here.
Right in the middle of the screen, we've got a pygnogonid or sea spider.
Can we see it on that screen?
Yes, you can.
Yeah? Great.
They're one of my favourites.
Yeah.
So
I think it should be said that even though there's no commercial fisheries here, we are fairly close to one of the heaviest fishing areas in Iceland. We are about just a few kilometers in shallow waters. So this highlights the necessity of protect habitats like this, because today there is no fishing going on, but that doesn't mean that in the near future it could be.
So should we start describing what we can see?
Yeah,
So-
I think it's good if we can, yeah. So we took a sample of this sponge yesterday. It's very similar.
Yes, it's an Antarctic, I guess.
Mm-hmm.
That's,
But this one, does it have a common name?
I think we sometimes just call it net sponge.
Well, to me it looks like a brain.
Yeah. So often we have common names as well as scientific names for these animals, where the common names are very descriptive and kind of describe what you see. So the corals that we see, we often call them cauliflower corals, or sometimes broccoli corals. Although sometimes we see them white, but obviously you don't get a lot of bright purple cauliflower.
No.
Well, can we find one of those? And
Just the pink, pale pink there. Pink one to the right. No, it's a bigger signal.
So this pink specimen here is a cauliflower coral in the middle of the screen.
sort of.
Yeah.
Well, common names here we can apply to lots of different creatures. So the little lattice at the bottom of the screen, can we see that one? Yeah. So this is called a lace coral, but it's actually not a coral at all. It's a kind of bryozoa. It's a different colonial animal. We took up some nice examples of these in a kind of tree form on some rocks a couple of days ago, and I'm sure we'll be posting them on our blog at some point soon. Can we zoom out a bit?
It's quite
Yeah,
unusual
yeah.
green color there.
Yeah, can we see the feather star?
Yeah.
Yeah.
Same here.
Here we can see a feather star, a crinoid. Again, the common name says exactly what you see. It looks a bit starry and feathery arms. Again, it's a filter feeding animal, but these can actually swim. And so sometimes when we disturb them or sampling, they swim off so that they don't get taken to the surface.
We've got a question regarding the colour of some species. So what's the advantage of being coloured in such a dark place?
So that's a question that we often get that used to stump me, but the answer that I give now is that a lot of these creatures, although we find them in the deep sea, they're closely related to organisms that we also find in shallow waters, and they don't necessarily lose their colour, but they're just relatives to shallow water species that do live in light where colour might be important. But also some of these creatures can actually produce their own light and bioluminesce. And we actually did some experiments with a sea pen a couple of days ago. We brought one to the surface, and they bioluminesce under under what's the word? So when you rub them. So,
So we agitated the stem.
Agitation, that's
we
the
tried
word.
with the ROV arm first. That did nothing, or our camera isn't sensitive enough to pick up the light that it emits. But in the lab, with all the lights off and basically in a cupboard, you can see it sort of luminesce slightly. Yeah.
Yes. So we had a lot of fun in a dark room, feverishly rubbing sea pens.
So if you want to be a marine biologist.
But here's a great example of one of the cauliflower corals, bright purple. Unfortunately, when you bring them to the surface and preserve them, they really lose their colour and turn into a kind of brown or creamy.
Whitish.
Yeah,
And
yeah.
that's the cauliflower.
Yes. Yeah.
We have a question about whether the exploration activities leave any impact on the habitat besides withdrawal of the specimens.
Well, this kind of work, like the one we're doing now, basically it doesn't leave any impact, except, as you say, for the specimens that we take. Other kind of surveys, like troll surveys, have a bigger footprint, but we try to make them, as minimum as possible. But that's one of the nice things about using an ROV with the manipulator tools like ÄGIR 6000 is that we can actually really be very specific about what we take from the seabed and really select the species that we want to obtain and try to avoid any additional impacts on the seabed.
That's a great segue into some sampling.
Yeah.
Yes.
So, should we try to do some sampling?
Yeah.
now?
So we've been working through a particular type of a bit of a list of specimens that we've had on images that we've not been able to ID, and this is the reason why we sample. These animals are extremely difficult to ID from images alone. We need to be able to look at their body organisations. So with the sponges, that's they have a particular type of formation using spicules. And also we use a DNA as well to characterise different species, and sometimes the morphology can be the same as another species. So now, in order to fully characterise the biodiversity in this area, we will take small samples from different species and we will bring them up to the surface and characterise them.
Yeah, and often, even if you have a specimen in your hand, you still can't reliably identify it. It really relies on very good reference material for comparison, and often you need a microscope. So the spicules that Emmy was speaking about are essentially tiny little shards of glass that the sponge used to make up its body, and you need to examine those under a microscope to look at the shape and the arrangement in order to correctly identify these things.
The period is moving.
Do we have any sampling requests here?
Well, we've been looking at the polymastids.
Yeah?
Yeah, so potentially we could take another one of those.
You mean not this one?
Yeah.
Yeah.
Should we use the
Which one?
vacuum?
Yeah.
Yeah.
This one.
The round
High-
sponge in the top, I think.
Yeah. Brown and yellow.
So this is where our expert ROV pilots really show their worth. But it takes a lot of training and expertise to be able to operate one of these things. Do we have a price tag on the ÄGIR 6000? How much money did it cost? 20 million.
So that's 2,000. 2 million.
2 million pound vehicle. So simple scientists like us won't be let near it. because we don't have the training.
We should say that we were like very pleasantly surprised about how easy it was for them to actually get the samples that we wanted. When we were planning this cruise, we were allocating a lot of time for sampling because we thought that it was going to be very difficult, but actually it has been fairly easy. So this is a very good thing, and that's because the training and the ability of the pilots.
Maybe explain a little bit about what's going to happen now, what we're doing here with the suction hose.
So you can see that. Can you see? Yeah. So the robot arm has picked up this hose, and the hose is going to essentially suck up a specimen and draw it directly into a containment tank at the back of the ROV, and so it's a matter of picking it up and pointing it in the right way, quite a lot like vacuuming. But obviously we want that to be very localized and only just to pick up the animals that we want to sample. It can be a slow process, but it's very deep water. It's a long way away, and it's very difficult conditions to work in.
What do you want to?
Bottom right.
Right.
Yeah. Stop.
Which one is it?
It's just below.
It's got the spikes on the top.
Zoom in a bit. Zoom in.
Yes,
Yep, zoom in.
Yeah. It's right under the hoover.
Yeah, top right hand corner.
If you go a little bit back. Yeah.
This one there.
Yeah.
Looks like a coral.
So once again, thank you for joining us live from the seabed while we are setting up the sampling. I just wanted to ask if anybody has any questions, please feel free to post them on the stream. We are trying to get to as many as possible, whilst also manipulating all the ROV equipment here in the control room.
And here we go. So this is, we think this is a polymastia.
Yeah, polymastid sponge. They're quite characteristic by the little points on top of them.
So the name polymastia actually means many nipples.
Alright. Are we gonna get it?
All of it quite.
It's in the long
We can try and pick up the rock otherwise.
Yeah, sometimes we need to scrap it from the rock where they're attached.
There we go.
Excellent. Yeah.
I think sponges are my favourite animal. I think everybody knows that though. Julian, have you got a wish list for anything that you would like to have seen or that you have seen so far?
Yes, I was actually really looking forward to see the umbellula, this sort of giant, what is the English name? Yeah, seapens. I've never seen them live in a video. This can be, they basically look like flowers and they can be up to 3 meters high. And we were lucky that a couple of days ago in Greenlandic waters, we found a place with very soft bottom, which is the kind of bottom that these animals like. And we saw some beautiful examples of umbellula.
Which container?
It's number 3.
Container 3.
So an important part of the process of doing the science is not just that we're not just here sitting and watching television, but we're also taking records of what we're observing and what we're sampling. so that we can tie things back. As I said, often when things come up to the surface, they don't look the same as when they're on the seabed. So when we're sampling here, we're keeping a record. We're putting things in different trays so that we can tie back the specimen in hand with the specimen that we've seen in the video, and in that way we're developing our catalogues of animals that we've seen, and we can then develop identification guides, and that's work that the guys in Iceland and Greenland have been doing to document and formally record all of the animals in the area.
I've got a question regarding sponges. Are they able to filter anthropogenic pollutants, and also are they affected by sea temperature rise like corals?
Filtering pollutants, anyone?
Well, I've never read anything about them filtering pollutants, but I think perhaps it's something that we just haven't looked at yet. And with regards to changing climate, I mean, obviously they draw silica from the seawater to build their bodies, so I guess if there is less silica available due to a changing the conditions, then that will affect the way that they grow and reproduce.
Yeah, also some sponges are very specific about their temperature that they like to live. So when we start to see changes in temperature in the seabed, then some communities will very likely to change. So yes, we are expecting to see changes because of climate change, and the kind of data that we're collecting right now will contribute to efforts in monitoring the seabed, because we don't only need to see it once, we need to keep an eye on these habitats regularly so we can see if they're changing or not.
We haven't seen much impact of litter on the seabed here. We've seen a couple of, I believe, fishing litter, but nothing too predominant, have we?
Yeah, no, we have seen very little. Litter. I think in part because we are far away from land, and there's not a lot of fishing activity going in this area. So that's why we are able to find these habitats in a very pristine and a very pristine condition.
So somebody's asked a question about whether we've been taking samples of the sediment and have we looked for microplastics. This cruise has been really focused on imaging the seabed and taking samples of any unknown specimens for us to characterise these habitats fully. So we haven't been taking sediment samples, but there are other cruises at the moment and expeditions on the Denmark Strait that are taking samples of the sediment for various different aspects. So I believe that some of that will be used to characterise whether microplastics have made it out this far.
Should we go for
Yeah. Can we stop at the Arctonian in the bottom there at the...
Yeah.
We'll sit down there so it's
Yes.
a stable picture.
Yeah.
So one of the uses that we're gonna, one of the objectives of the cruise is to generate the data that later we're going to use to map these habitats, and that's a very important thing that we do during this type of activities. When we go to the seabed, we can only see a little bit of the seabed every time, but we need to know how far these habitats extend. So there's will be, afterward when we go back home, there will be a lot of work analyzing the data and doing these predictive models of the habitats, so we can actually create maps of the area, and we can decide about protection measurements based on the extension of the habitats.
Yeah, I mean, it's a good point that although we're getting really awesome pictures here, we might do a transect, and that transect might go on for a couple of hours, and we might cover 500 meters to a kilometer at most, and that's gonna see a tiny speck of the vastness of the seabed here. So we can't ever hope to visualize the entire seabed, and here we're going to be at sea for 10 days. We're going to conduct 20 or 30 dives. You know, so that's going to give us 20 or 30 kilometers of transect, but and that's covering an area of many thousands of square kilometers. You know, a tiny fraction of 1%.
Yeah.
Even the most basic seabed observations, detailed seabed mapping, which is something that we can do from the ship without a camera, using multi-beam echo sounders. We can kind of go up and down and essentially do a kind of lawnmower type surveys where we're measuring the depth of the sea below us. But even that most basic survey in Iceland, only 30% of the waters have been fully multi-beamed. I think David told me yesterday. And they have a 10-year program going at the moment, and that won't be finished at the end of 10 years.
No.
Just to clarify, people watching might not understand or know what multi-beam is. So can you quickly just give an overview of how we conduct mapping in the ocean?
Yeah. So when we do this, the first thing that we need to do is to map what we call the topography, or the shape of the seabed. So so for that we use an acoustic equipment called a multi-beam echo sounder, which is basically, it's like I like to imagine it as a fan of flashlights, but instead of showing light to the bottom, they emit sound, and the sound bounces on the bottom, and that gives you a measurement of how deep the seabed is at that place. So when we start to going back and forth with the ship, we can get a continuous map of the seabed with a lot of detail, actually, on how deep or how shallow the area is, and also if the seabed is hard, like rocky, or if it's soft, so if it's made of sand or mud. And that's a very important first step, but that doesn't tell us anything about which kind of animals do they live in that area. So the next step after that is to come out with cameras and try to list the species that we see in every place, and then trying to identify which communities or which ecosystems they belong to. And then the third step is when we have to just put all the information together to create these maps. And that involves a lot of statistical analysis and modeling work, because as Chris said, we're only watching a tiny little bit of the seabed, and it's quite challenging to make a complete map. I like to imagine, like, if we want to make a map of Iceland or the UK or Greenland from an airplane, and just flying up top and just taking a few pictures here and there, and then you want to make a complete map out of that, so it's very difficult. But it's an essential tool, because we cannot protect things that we don't know where they are and how long, how far they extend. So that's why several programs in Iceland, in the Marine and Freshwater Research Institute, and Greenland also, and many other countries are doing these habitat mapping programs to basically produce maps of the types of communities and ecosystems that you find on the seabed.
I've got a question about the field of view that we've got with the various cameras. I mean, there's 4K camera, we can zoom in and out, but I mean, do we know what the average field of view is for the HD camera, which is the main view?
That's a question for our ROV technicians. The field of view of the camera, how far? It is.
Well, I think we'll have to
70 degrees. Something.
Yeah. 70 degrees field of view.
Yeah, this one here.
Now you can see us picking out the sample and putting it into one of our sample trays. It's very delicate work.
Somebody's asking, can we estimate how old these sponges are? Well, I guess probably from images not, but I mean sponges grow very slowly. A lot of the species here grow very slowly. So, and this is a very established community. So I wouldn't like to hazard a guess, but they're not new.
Yeah, it's a very difficult group to age.
Yeah.
There's really much that you can rely on.
No, there's no real method to age a sponge that I'm aware of.
Yeah.
With other organisms such as corals, you can use techniques such as carbon dating on the solid body of the animals, and that's been used on many corals to show that they can live to beyond thousands of years.
Yeah, in some corals, they also have like a year rings in the stem,
Yeah.
like trees.
Yeah, they have rings like trees.
Yes.
Yeah.
So that can be used also as an indicator,
Mm.
but that doesn't apply for all of them. It's just some types of coral trees.
Yeah.
But one way that we can have an idea is that when we put man-made structures in some places, or where there's for example, a ship that went down, then we know that there's a brand new surface, and then if we visit sometime later, we can actually measure and get an idea of how much those animal sponges have grown since. And that type of work has been done, for example, in places where they put oil platforms or pipes, but that will give you only like a very vague estimation of the age of those animals.
Well, we tried to look at a platform that had been deposited 20 years ago yesterday, but we couldn't find it.
Yeah.
I suspect it's probably been grown over so much that you wouldn't have been able to see it.
Yeah, maybe, or maybe it's just it's a small object that it was hard to find.
Yeah.
Yeah, the seabed is a big place.
So now we're going to just go off on a little bit of a wander and or conduct a transect, as we like to call it.
Bit of seaweed there.
Yeah, so it's amazing. We're more than 100 kilometers offshore and 600 meters deep, and we still see bits of seaweed that have sunk and been washed across to the seabed here.
So what other wildlife have we seen so far apart from the invertebrates that we've been focusing on?
Well, we've seen several types of fish. They tend to hide or escape because I guess the ROV is a big and bright thing that scares them away. We've seen also a lot of shrimp-like animals in some places. In some situations where we have these, like small shrimp that get attracted by the light of the ROV, more or less in the same way that moths get attracted to light inland. So in some places, you have a lot of movement on the screen because of that.
So this kind of very structurally complex habitat is a really good place for lots of small mobile animals to be able to hide themselves and go and lay their eggs, and that's one reason why it's important that we document and try to conserve these habitats, because these are really important nursery grounds for many fish and crustaceans and many of the things that we like to fish and eat from the sea.
We had a question about all the echinoderms that we can see, so the brittle stars, sea stars, and I'm not sure if I've seen any sea cucumbers, but yeah, I mean, it's really difficult to ID them from images, but they obviously are aggregated within these high density sponge grounds because there's a lot of food here, right?
yeah.
We've got a question about the camera. So what kind of cameras do you use on the unit? The pictures are very clear. They are.
Oh well, it's good to hear that that the stream is coming through clearly.
Yeah.
That's been a bit of a worry for us. So the smaller picture-in-picture camera is a 4K video, so super high definition. It means kind of like we very quickly rack up terabytes worth of video files by playing it, but it also means that when you play it back, you can zoom in closer. Sorry, you can zoom in really close and get a really good eye on the detail.
Oh, that's lovely.
The main camera is a high definition camera. I don't know what resolution that is. Standard high definition camera, yeah. Which itself is still pretty good.
Yeah.
Certainly a lot better than the homemade GoPro cameras that we've been using previously in Greenland.
But it works.
Yes,
It works
very well.
It works.
Yeah.
It works really well. But you get what you pay for. £2 million buys you a lot of really good quality imagery.
Yeah. Yeah, we are not used to get imagery of this quality in our surveys either, and it has been, yeah, a source of wonder for the last few days.
Yeah, it's been very difficult to go to bed when you're off shift, because when the other team takes over and you feel like you're going to miss out seeing an amazing habitat. We can stream these from our cabins, so a lot of us have been going to sleep with them playing in the background.
It also happens that sometimes, we've been on the seabed in a particular beautiful location. It's time to go up and go to the next place, but we don't want to. We want to keep watching and keep discovering the place.
So somebody's asking, there don't seem to be many mollusks or crustaceans visible. Is that to surprise?
A lot of the mollusks are quite small, and so they can be quite difficult to see. We have seen a few.
Yeah. We have a lot of squat lobsters, and they've been hiding under rocks and within sponges.
We can see an empty shell here. But yeah, I haven't seen as many squat lobsters in this station as we had in some of the others.
But there are a few small, tiny amphipods that are crawling on the other animals that you can sometimes see.
Well, so far we have taken, I think, 16 or 17 ROV dives in this cruise, and the diversity of the habitat has been really surprising. We have been, there are very clear differences in the type of animals that we see in different places, and that has been a very interesting part of this research.
You can see here that we've gone from that patch of brightly coloured, highly diverse organisms to a much plainer gravel bed. It kind of shows the dependency of many animals on the seabed type, but it also shows how patchy and these habitats are. We can go from really dense sponge ground to pretty flat gravel bed in the space of a few meters, and that makes the job of modelers like Julian all the more difficult.
Yeah. Also, because there's still a lot that we don't know about what are the physical and chemical properties of the habitats that influence the distribution of different animals. Another interesting thing in this type of habitat is the importance of rocks. In places where you have more soft sediment and you find isolated rocks, rocks are almost like islands of life. And they play a very important role in supporting communities of sponges and corals. And you can see here how they're colonized by a bunch of different animals. And sometimes when we sample, it's easier just to grab a rock that is full of animals rather than trying to pick the organisms by themselves.
You can see here though how you have some things growing on top of other things, you know, so you get this kind of little tower of organisms all living on top of each other.
Are those polychaetes, little worms?
That's a spider.
He's really cool.
Should we have a bit of a transit now, and maybe that'll increase our chances of seeing some fish?
Yeah.
I imagine we've scared away all of the fish with sitting quite stationary for a while.
Somebody said the purple is so vibrant, and yeah, in this particular habitat, we've noticed that these soft corals are really vibrant here, and it's really a pleasure to see for us, and such a healthy and vibrant and dense community. So what do you enjoy most about being at sea?
The company of the people, the food.
Good answer.
Food has been really good. And the having the opportunity to see these habitats. To me, this type of research is it's like exploration in the most basic sense. We are actually seeing places that nobody has seen before, and that's always very exciting to me.
What about you, Chris? What's your most favourite part about being at sea?
Yeah, I'm going to have to mirror Julian's answer a bit there. Just certainly this cruise, the quality of the imagery has been really amazing, and it's been great to get such close and detailed view of the animals that we've seen a little bit more at distance previously.
But this sort of paraphrasing Star Trek, this is the seabed, the final frontier, and we are going to places that we have not seen before.
Oh, we've got one here. My father Jonas is driving the ROV. I think the sea creatures are very cool. / 4-year-old Scott. Aww. Got a shout out. Well, we're really glad you're enjoying it, Scott. And your dad's looking after us.
Yeah, he's doing an amazing job.
Yeah.
Yeah, I think I should extend that to the whole crew of the ship.
Absolutely.
Yeah, they have a a very high level of expertise and professionalism, and they have catered to every one of our whims and needs. We want to go here, we want to go there, and they have taken us with a very high level of precision, and yeah, we're very thankful for that.
So we have a question saying that one of the viewers thought they saw a conch. Is that possible? I didn't realise that we had. Well, some of the mollusks look quite similar, but I'm not really sure that we have an ID for these, do we, Simon? A conch is small.
Is that what I call a conch snide?
Yeah.
One of the big smells that you can blow.
Yeah.
No, that's, no.
Yeah.
Must be something similar.
A lot of the mollusks in this area aren't great. Haven't been hugely characterised to my awareness, but yeah, we'll probably do some work on the images once we've got them back and we're back on dry land to try and understand what's here. Another question here about how long are we away at sea for and what's the food like on board? Who's the best cook? The food is amazing.
Yeah.
We get cake at 3 o'clock every day, which is the best
Some
time.
of you are getting cake at 3 o'clock every day.
Unfortunately, some of us have to work night shifts, and this time round, Nanette and Laura and Berger.
We are
sorry for
missing out
pronunciation.
on the cake.
I'm missing out on the cake because they're sleeping.
Not today, maybe.
Yes.
Not today.
No, that's help us.
Yeah, well,
Yeah.
and actually not yesterday either, but that's another story.
I promised to get them cake and I forgot. I apologise and I'll get it tomorrow.
But the truth is that food is really important in any ship, so I could, it helps you to lift your spirits. It's not only nutrition but also, yeah. So it has been really, really good here. Yeah.
So we're coming up on the hour of streaming, so we were aiming to stream consistently for around an hour today. So if you guys have any last questions to ask us before we wrap up, please post them in the comments and we'll do our best to get through them.
Really beautiful picture.
Amazing animals.
A small one.
On
Oh,
the
that's
on
small
the
one.
Yeah.
Chris, your wife's saying don't trust him to deliver the cake.
Thanks.
Lots of people saying amazing images and really nice habitats. Thank you all for your positive feedback. We are so excited to be here and so thankful that we've been given this opportunity. Anybody that works on deep sea images will know how difficult it can be. So these have really given us a unique view at quite, truly mysterious environments.
We are very happy that we've been able to do this, and thank you, Evan and Chris, for organizing all this live event.
Well, it's an absolute pleasure.
Big thanks to Emmy for that
Yeah.
and all the work that she's done.
Yeah, she was amazing.
Are you going to talk about the Eurofleets stream, or we're
Yes,
hoping?
yeah, we're going to do a couple of streams on Monday from the Eurofleets page, so we'll post a link to it in the chat as well and in the event page. It won't be hosted as it has been today, but if you're interested in just watching us in watching us do a few transects, then we'll be streaming the video live to you as long as the technology and the weather holds out.
So now we're watching something different.
So this is something different, and we will hold off so we can have a look at this a bit more closely. This is not something that I've seen before. It's quite an enemy-like.
Yeah.
Oh. It looks like it's lost a few fingers.
Yeah.
Couldn't it be egg-sacs?
I think, yeah, they look like eggs.
Could it be what?
Or like
Egg-sacs?
eggs from squid, maybe? Squid.
So this potentially could be squid egg sacs.
That's beautiful.
So here we have it. We can look at deep sea
You wanna
videos
zoom in and get a
all
picture?
day long, and
Oh, yeah.
we can see something that we've never
Well, we
seen
can
before.
video
Yeah.
doing it. Wow.
Yeah. Yeah, they're eggs.
I think that. Yeah.
Right? So I have to take a sample.
We're going to get the vacuum out.
Yeah, should we
So
start
we're gonna
with
take
it?
a sample with the vacuum.
Yeah. Yeah, let's take a little bit of that.
Yeah, just a small part.
So I think we'll wrap up the audio here for now. So thank you all for joining us, but we'll keep the live stream video on so you can see a sample, this quite unique object on the seafloor. We've got some speculation that it could be some kind of egg sac, but we'll keep streaming the video for a few minutes or so you can keep up with us. But we will say goodbye from us.
Yeah, goodbye.
Goodbye.
Thank you for joining us, and it's been a pleasure.